hit that I have done this and not recorded a lecture. Okay. Is it recording? Does it say recording? Nope, it is recording. Okay, cool. Let's give this bad cat a shot. All right, folks, 2 extremely crunchy chapters. I will tell you that when I get feedback from folks about the course, they're always like, oh, you were trying to kill us with that cellular respiration and that photosynthesis. And students often tell me, good morning, often tell me, what's your name again? Tell me your name? Sorry? Kyla. Okay, great, Kyla. I'll just mark this here. So they often tell me that they don't like the photosynthesis part. And I am firmly convinced that that's part of our bias. We tend to be biased toward animals. I know I am. I tend to kill plants. That's like a hobby of mine. I try to grow them and then I kill them. But if you understand cellular respiration, then you are going to understand photosynthesis. For those folks that have watched the lectures, do you see the connection? Okay, and if you haven't, think about those connections as you're going through. So let's kind of review where we are. Remember that we are still talking about cells, right? I'm gonna do my best not to write like a crazy person. And we're in this area where we're thinking about how cells function. And so these 2 chapters together are really going to tell us something about how cells process energy. This is working better than I thought it would. Okay, so basically, I want you to think of this as, okay, we're gonna have like a 2-pronged, how do I wanna think about this? Okay, we're gonna process energy. We're either going to consume chemical energy, yeah, that's right, chemical energy. And if you're consuming the chemical energy, then you're using either cellular respiration, which I'm going to abbreviate, or you're using a process called fermentation, right? What is the chemical energy that you would be consuming in either of these? When you're consuming, like at the most basic level, like we tend to consume a variety of things, but in general, what'd you say? ATP is actually, I'm glad, put a pin in that, okay? We're gonna come back to ATP. Yes, you are consuming that. But it turns out that to get that ATP, it's almost like you have to exchange money. If you ever travel to another country, you got money in the bank, right? But if you go to England, they use pounds. So you have to convert your your dollars into pounds to make them useful. But you've got money. I want you to think of this process in the same way. So ATP is what you can actually spend, but what's the thing that you have in the bank? Glucose, yes, okay? So you're actually going to take glucose in these examples, and you are actually going to exchange them for ATP. Are y'all with me? So we're going to be using up glucose and we're going to be turning it into ATP. That is the goal of both of these processes, okay? Now, here's the deal. This is consuming the chemical energy, but somebody has to store or create the chemical energy, right? So we'll put, I don't want to say create because you can't create energy according to the laws of physics, but we're just going to say store chemical energy. And can y'all guess what process does this? Yeah. And so I'm going to abbreviate photosynthesis as PS, right? So photosynthesis is taking the light energy that you have from the sun and turning it into that chemical energy in the form of glucose, right? So some concepts that I want you guys to remember as we go through all of this, okay? I want you to remember a few things as I teach you this stuff. First of all, remember that electrons are going to equal energy as we go through this process, okay? I want you to remember that. We're going to talk about oxidation and reduction reactions. How many of you guys had this in high school chemistry or you're having it in chemistry right now? How many of you hate this to the bottom of your gut? Okay, well, in biology, I'm not a chemist, and we're gonna keep this super simple for you guys, okay? So you are going to need to understand oxidation and reduction. And here's why. It has to do with electrons. And what did I say electrons equal? Energy. So when you're thinking about oxidation reduction, I want you to remember this mnemonic device, oil rig. Anyone that's ever had chemistry, can you tell me what oil rig stands for? Caleb, you've had it. I know you have. I was like, ah, crap, I was afraid she was going to call on me. Is? Yes. Boom, that's exactly right. Did y'all hear that? Oxidation is loss. Reduction is gain, okay? So if something is oxidized, it has lost an electron. If it is reduced, it has gained an electron, and you're thinking, that is stupid, because if you reduce something, you're going down. But why is it that way, Caleb? No, we're not going to go into that. We're not going to go into oxidation states. We're not going to do, because that is, we're keeping it very simple for biological purposes. Here's why you need to know this. Okay, the reason it's like reduction is gain is because you're gaining an electron, but that's, it's just, it reduces the charge because the charge is negative. Don't worry, just remember oil rig and you'll be good to go. But the reason that's important is once something has been reduced and it has gained an electron, y'all, what did it gain? Energy. So we're going to be using that, okay? The other thing to know about this, there was something else I was going to tell you. Oh, crap. Why did I want to? I lost my train of thought. I'll come back to that in a second. All right, the next, so we've got these things right here. We're going to need, oh, I know what I was going to tell you. Also, in biology, when we're talking about redox, oxidation reduction reactions, we're going to make it really simple. I'm going to tell you guys to follow the hydrogens, follow the H's. Because remember that a hydrogen is a proton plus an electron. So be watching things. If something gains a hydrogen, it's gained an electron, it's been reduced, and it's carrying energy. Are you following my line of reasoning? Okay, the last little concept, these are all things that we're going to see over and over again in this chapter. The last concept is this idea of gradients. Y'all remember when we talked a little bit about cell membranes, and we talked about setting up gradients, diffusion gradients. So you have some things go from an area of high concentration to low concentration. And y'all think about that in terms of chemical, right? But remember that there's electricity can follow a gradient too. So you actually call these gradients electrochemical gradients. And we're going to use these gradients today. Electrochem gradients. Okay, so these are the things that I want you to kind of bear in mind as we go through our little discussion today. So let me switch to a new clean page here. All right, so let's start with our idea of consuming chemical energy. So we're going to talk about cellular respiration today because that's the harder of the two. In your chapter, you are responsible for cellular respiration and fermentation. I'm going to leave you guys to, because I only give you a little bit of information about fermentation. For today's purposes, here's what I want you to know about cellular respiration versus fermentation. Cellular respiration, which is what I'm going to talk about in a second, is what we call aerobic. So can you guess what fermentation? Yeah. Now, what does that mean, y'all? Excellent. And there we go. I need an R, sorry. Okay, so that is without oxygen. Why is that important? What you're going to see is that if you have oxygen present, you can make a crap ton, scientific term, a crap ton more ATP, bless you, than you could if you didn't have oxygen present, okay? So that is one theme that you need to be aware of. That's something that I might ask you on an exam. If you are doing aerobic respiration, you are able to take your molecule of glucose, karate chop it up, extract some energy from it, and turn it into ATP, and turn it into a lot more ATP than you could if you didn't have oxygen present, okay? So let's talk about that. what we're going to do is do one of my giant charts that I like to do. I don't know if you guys find these helpful, but I sure do. All right, so here's we're going to make giant chart here. And how do I want to divide this up? I think I want to divide this up thusly. So what I want to focus in on right now is cellular respiration. Now, here's the thing you need to know about cellular respiration. All cells are doing this all the time, okay? Because it turns out your body doesn't do a very good job storing ATP. You only have enough ATP in your cells and in your body at any given point in time to do about 30 seconds worth of chemical reactions. You consume that stuff real quick, okay? It's kind of like, you know how my analogy is spending money, like, okay, you've got these dollars and you've gone to London for an awesome vacation and you need to turn these dollars into pounds, right? But if you're like me and you've got cash in your pocket, that cash is gonna go really quickly, right? Because you're like, hey, what's happening? Okay, so this is the same kind of idea. So every single cell in your body, in any organism's body, is undergoing cellular respiration all the time. If it's an aerobic organism, I say that because there are bacteria that can only do anaerobic respiration, okay? So here's a question I might ask on an exam. Do plants undergo cellular respiration? Yes, people say no all the time. What plants are doing is they're harvesting the energy from the sun, they turn it into glucose, but then they karate chop that glucose up to turn that into ATP because not every part of the plant is facing the sun, right? You got to send energy to the roots. So yes, plants do both. They photosynthesize and they do cellular respiration, okay? Now you can think of cellular respiration as occurring in 4 basic parts. The first part, and this actually is part of fermentation too, is called glycolysis. The second part is pyruvate oxidation, and I'm going to abbreviate that oxidation, it's an OX. The next part is called the citric acid cycle. You might have heard of this before as the Krebs cycle, or sometimes you'll hear it called the TCA cycle, which stands for tricarboxylic acid, okay? Those are all 3 the very same thing. And then the last part of cellular respiration that I'm gonna make you guys learn a little bit about is, I'm gonna abbreviate this ETC for electron transport chain, but there's more than one kind of electron transport chain. So another part of this another terminology that I want you to know is the term oxidative phosphorylation. And the other thing that you might hear is chemiosmosis. And I'll explain all this in a momentito, okay? The osmosis. Okay, so let's start here, y'all. Glycolysis, step 1, okay? The word glycolysis, if you are to break it into its word roots, it literally means glyco, sugar, lysis, break. We've had that term lysis before, and I think we've had the prefix glyco before, okay? So in glycolysis, you are breaking a sugar. What's the sugar that we are interested in, y'all? Glucose, exactly, okay. So glucose is a 6-carbon sugar, so I'm just going to draw 6 little circles. And I'm going to draw bonds here so that you know that those are carbons that are bonded, okay? So that's my representative of a glucose molecule. Are y'all with me so far? What glycolysis essentially does, I am not interested in y'all learning enzymes. Some professors want you to learn enzymes. Let me tell you something. I got a PhD and a bunch of scientific publications, and I can't tell you the enzymes of glycolysis. If I need to know that, you know what I do? I look that crap up, okay? So I do want you to know the gist of what happens. So in glycolysis, we're gonna break this sugar, and we're gonna break it into 2 3-carbon molecules. We have lost 0 carbons at this point, but we've changed this, haven't we? So it's just like the box says, glycolysis, breaking sugar, we broke the sugar. Now, in the process of doing this, and I do go into a little bit more detail in the video. So that level of detail is what you need to know. I'm not going to ask you to know a bunch of enzymes, but I do want you to know that in addition to these 3 carbon sugars, which by the way are called pyruvate, in addition to that, you're also going to get some other byproducts. You're going to get 2 ATP, okay? And that ATP comes from adenosine diphosphate that gets added to another phosphate group. So there's a chemical reaction where you take ADP and you add another phosphate on it and you get ATP. So that's one byproduct of glycolysis. A second byproduct of glycolysis takes a molecule that has a whole bunch of carbons on it. It's a molecule called NAD, and I can never even remember what NAD stands, like nicotinamide, adenosine diphosphate. Don't worry, NAD is all you need to know. But NAD is a big molecule that we can use to stick a hydrogen on. We're going to take NAD and we're going to turn it into NADH. Y'all, what did I just do? We're actually going to get 2 of these, by the way. Can y'all see that I added a hydrogen to this guy? That means that now this guy is an electron carrier. That means that this guy is reduced. That means he has energy. Y'all following me? Okay, so that's what you get out of glycolysis. Next step. I want you guys to just kind of ignore these guys for a second. We're going to follow these guys. These guys are now going to move from where they live in the cell. This part happens in the cytosol of the cell. And these guys are going to move to another location of the cell. Remember what the goal of this process is. The goal of this process is to take sugar and turn it into ATP. With that knowledge in mind, would you like to guess where these guys are headed? Boom, because it is the powerhouse of the cell. Thank you very much. So we are going to the mitochondria now. Okay. Okay. Now, when we get to the mitochondria, these 2, bless you, my allergies are acting up too. I'm sneezing all the time. Okay, so these 2 3-carbon molecules are going to get changed a little bit. They are going to be added to another large compound, and then we're going to have 2 carbons. So draw them like this. So we're going to end up with 2 molecules of something called acetyl-CoA. This box is the CoA. It stands for coenzyme A, and it's kind of a beast, so we're not going to draw it out. But do y'all notice anything? Tell me what you notice. So I've got this box. We added this box. We just added this part here. Here's our little carbons. So we went from these guys to these guys. Did anything change besides the addition of the CoA? We lost 2 carbons. Y'all see that? So we added the CoA and we lost 2 carbons. We lose those 2 carbons in the form of CO2. What do you exhale? That's right, CO2 is carbon dioxide. You exhale carbon dioxide. When you exhale, you're getting rid of the waste products of cellular respiration. Does that make sense to you guys? Okay. Now, additionally, this process is also going to generate 2 NADH molecules, 1 for each of these guys here, okay? So again, we see this hydrogen is here. This must be an energy carrier because it's hanging on to an electron. Are y'all with me so far? Next step. This cycle, some biology teachers really insist that their students learn all the steps of the citric acid cycle. I am not one of those professors, okay? I just don't know that I think that in your nursing program, you're gonna need to know that, or if you do, you'll learn that in some other context. I'm more interested in you understanding the concept, okay? So what's gonna happen next is something called the citric acid cycle, and here's why it's called a cycle. It starts out with a 4-carbon molecule called citrate. See where the name comes from? And then we're going to take to that 4-carbon molecule and we're going to add our acetyl-CoA and end up with 6 carbons. Are y'all with me so far? 1, 2, 3, 4, 5, 6. You see how I did that? So we took these 2 carbons from acetyl-CoA and we added it to this guy. Now, there's going to be a series of different chemical reactions that are driven by enzymes. And what's going to happen throughout those chemical reactions is this 6-carbon molecule is going to be manipulated, and at the end, you're going to end up with a 4-carbon molecule again. Yes, ma'am?
And that happens twice, right? Because you have 2.
Yep, put a pin in that. We're going to come back to that. You're absolutely right. So we're just taking one of these guys here, and we're adding it to this. Is everybody with me so far? Yes, ma'am. Okay. No, that's great. No, I'm glad you asked. Okay, so we started out with this. This is just hanging around in the cell, this 4-carbon molecule. This 4-carbon molecule hangs around in the cell to participate in this cycle. What we added, Janice, right? Janice, what we did is we took this guy here and we inserted the 2 carbons from this guy onto this. So you have these 4, 1, 2, 3, 4, and then these 2, 1, 2. You with me? Okay, excellent. So there's going to be a series of manipulations, and at the end of this whole cycle, you end up with this original 4-carbon molecule. But what have you done? You've peeled off 2 CO2, right? Because you see how I went from 6 to 4 again? So that means I lost these 2 at some point throughout this chemical cycle. You also gain some other byproducts. You're going to gain some ATP, 1 ATP per turn. You're going to gain 3 NADH. What did I tell you guys about this guy? It's a what? Electron carrier, energy carrier, yes, okay? And here's a new one that I haven't introduced you guys to. You're going to gain a molecule of something called FADH2. See how it looks similar to this one? Also an electron carrier, okay? So for each turn of the citric acid cycle, you get 2 CO2. This is waste. You blow that out your mouth, okay? You get 1 ATP, 3 NADH, and 1 FADH2. But as Jennifer pointed out, each turn of this citric acid cycle corresponds to one of these guys, right? So if we're going to chew up a molecule of glucose, we're going to turn that cycle 2 times. Do y'all see that? Okay, so that means that you can multiply these products times 2. Are y'all with me so far? Okay, now let's kind of do some math for a second. We lost 2 carbons in the form of carbon dioxide right here, right? I'm going to actually do this. Hold on. Let's highlight it. So we lost 2 carbons here, right? We lost 2 carbons here, but we did it twice. How many total carbons have we lost at this point? 6. How many did we start out with? 6. We've chewed up our glucose at this point, is what I want to tell you. Okay? We've chewed up our glucose. So the next part, let me get rid of that. No, wait, I just don't want you to do that. There we go. Bless you again, my goodness. Okay, let's see if I've got, yeah. So now we've chewed up our glucose, and the next phase of cellular respiration is going to be all about our energy carriers. These energy carriers are going to take their sweet little electrons, and they're going to use them in an electron transport chain. Bless you again. You've just got some allergies happening, don't you? The electron transport chain happens in the mitochondria, and you know, I'll draw the mitochondria for you in a second, but it happens in the mitochondrial membrane. Just know that, okay? Here's what the electron chain is going to do, electron transport chain is going to do. We're going to go over this in a little more detail in just a second. But things that you need to watch out for as I go over this in more detail in just a second. It is going to use oxygen, okay? The reason it's going to use oxygen, we're going to be stripping these electrons here off of these carriers. And we're going to use that energy to drive enzymatic reactions. Do y'all remember, bless you, oh my gosh, the pollen. Do y'all remember last week when I was talking about energetic coupling, how you can use a reaction that gives off energy to drive an energy consuming reaction? These electrons give off energy. We can use that to drive energy consuming reactions. That's what we're going to be doing, okay? But when we're done, we're going to have these, like a handful of electrons. We got to put them somewhere. Once we've used up their energy, we got to dump these electrons somewhere. The somewhere that we dump them is onto oxygen to make oxygen gas, okay? You have in your cells oxygen that's lacking electrons and another one that's lacking electrons. You dump some of these electrons on here, you form oxygen gas. This is why you breathe in oxygen. You breathe in oxygen so that you have a place for these electrons to go. You exhale carbon dioxide as the waste product of glucose. So the whole reason, we talk about respiration, you think of breathing. Do y'all see how they're related phenomena? You're breathing in to get the oxygen, you're breathing out to get rid of the waste, which is carbon dioxide. Pretty cool when all these things come together, okay? So you're gonna use oxygen. This process is the one that makes ATP. This is where the majority of ATP gets made. So far, we had 2 ATP that we made here, and we had 2 ATP that we made here, but that's a measly 4 ATP. We're actually gonna be able to get 38 ATP out of a molecule of glucose, and that action happens in the electron transport chain, okay? The last thing that the electron transport chain is gonna do is it is going to generate, so I'll put gen, water. It's actually gonna make something called metabolic water. It's not sufficient for you to not drink water. You can't be like, well, I'm making water, so I don't need to drink water. No, that's not true. But you do make a small amount of water metabolically, okay? Questions before I go on to the electron transport chain?
Any of this cost energy?
A lot of it costs energy. And so for example, I think there's an energy investment phase and an energy harvesting phase here, okay? This stuff I think does not cost energy, but all of this over here that happens costs energy, which is why you're using your electrons from your high energy electron carriers. So some of it does and some of it doesn't, okay? So when you actually watch the video, you'll learn that in glycolysis, to get this party started, you have to give it 2 ATP. You actually make 4 ATP, but because you already spent 2, we say that you get 2 total out of it. Does that make sense? So y'all need to watch that part. Another question? Question? No? Okay. All right, so let's move on and talk about the electron transport chain. This thing is working okay. Do y'all like this better than turning back and forth to see the board? Yeah. Okay, then I'll just start bringing this to class because I wasn't sure how this would work. So I'll just bring this to class. All right, y'all, we're gonna draw like we do, okay? So first off, I want to draw you a mitochondria. Y'all are familiar with the fact that the mitochondria is an organelle that you find in eukaryotic organisms, right? If you were a prokaryotic organism, you could do cellular respiration, but this would all happen in your cytosol or in your plasma membrane, okay? So mitochondria look something like this. If you ever see them drawn like in a cell diagram, they look like wonky jelly beans. Have y'all seen this before? They look like weird jelly beans because they tend to be oval. They have an outer membrane. So I'm just going to abbreviate OM. Now, this membrane that I drew here, I drew a single line, but remember, it is a phospholipid bilayer. So if I was drawing it right, it would have the little head and the tail and the head and the tail, right? But that's a pain in the butt. We're not going to draw that. It also has an inner membrane, which I am just going to abbreviate IM. Again, remember, that is a phospholipid bilayer. This inner membrane, outer membrane configuration, Chapter 7, I tell you guys about something called endosymbiotic theory. If you watch those lectures, you should know what that is. I'm 99% sure that I've written a question about that for your exam. I'm not 100%, because that might have been last semester's exam. I'm in the process of writing your exam, but I usually ask about endosymbiotic theory. Do not be surprised if that comes up on this exam as well, okay? So you have an outer membrane, you have an inner membrane. This area here is often just called the inner membrane space. And then this part on the inside here is called the mitochondrial matrix. Now, why do I bring all this garbage up? Well, remember how we said the mitochondria is powerhouse of the cell, right? And we moved our pyruvate into the mitochondria. So the Krebs cycle and making acetyl-CoA, that stuff happens in the matrix. So it happens in here, okay? Y'all with me? The electron transport chain, though, it happens, it's a series of proteins that are embedded in this inner membrane. And that's why this inner membrane is all wrinkledy and it's all like tore up looking, because what you're doing is you're creating maximum real estate, maximum space to crowd these electron transport chain proteins in here, because the more electron transport chain proteins you have, the more of this you can do, the more ATP you can make. So can you see how this is an adaptation to maximize space for these guys? All right, so let's draw this electron transport chain. We're going to first draw an outer mitochondrial membrane. So I'm just going to draw it thusly, and I'm going to label it OM. So this out here would be the cytosol of the cell, okay? Are y'all with me in terms of where we are? Then I'm going to draw for you here the inner membrane, okay? Maybe. Okay. Inner membrane, great. And then this down here is the matrix. So we're kind of like, it's like I took a chunk right here, okay? Now let's draw some proteins, and I'm going to keep this real simple. We're going to draw these protein complexes as boxes, okay? So I'm going to draw in a box here. I'm going to draw a box here. Let's see, I want to draw one here. Whoops, come on. One here. And then we're going to draw something that looks like an upside down Christmas bulb, like from Christmas lights. Okay, can y'all see that that was kind of what that looked like? Everybody good so far? Here's what's going to happen. So all of our high energy electron transport, all of our high energy electron carriers that we made, all of our NADHs and our FADHs. Remember how I said that all this stuff was happening in the matrix of the mitochondria? So here they are hanging out. Now, what these different protein complexes do is they're able to strip the electrons off of NADH so that you end up with NAD, and you have an electron here. Are you with me so far? We've stripped that electron off, right? Now, we are going to use the energy of that stripping off. Energy is given off when that happens. We're going to use that to do something cool. Remember how we couple these energy giving off reactions with energy consuming reactions. We're going to use the energy given off by stripping this off to pump hydrogen ions from here to here. Nope, sorry. I have this drawn a little bit wrong. It shouldn't be here, it should be here. Sorry. I should have drawn these down here. Y'all, I'm sorry, correct your drawing, because where we're going to end up with our hydrogens is in this inner membrane space, okay? Can y'all correct that? You see what I did wrong? It's not actually going to go out here because they would float away, okay? We don't want them to float away. We're actually going to capture these H pluses, okay? So here we go. We've got an H plus that we've used, okay? Now this electron still has some energy. So we're going to hitch this electron to basically like a shuttle bus molecule called Q, okay? That stands for ubiquinone, but don't worry about that, okay? And I'm just going to color that in because this is getting a little bit messy. So this colored in thing is called Q. But don't worry too much about that. I'm not interested in names. I'm interested in concepts. This is a shuttle bus. What this molecule does is it allows this electron to attach and it transports it over here. We're going to skip one, okay? We're going to skip this one. You'll see why in a second. So now we're going to move this electron over here. So now here's our Q. Our electron is going to get off the bus. We've moved this electron around. Guess what? That is still some energy that we can use. We're going to pump hydrogen again into this space. Y'all see what I've done? Okay, now we're actually going to use a different molecule this time. It's called a cytochrome molecule. Don't worry about that. It's a different shuttle bus, so I'll draw it as a circle. Okay, different shuttle bus. But basically, it's going to take this electron one more time over here, and it's going to drop this electron off. Okay, so there's our shuttle bus. Our electron got dropped off. We moved an electron around, so can you guess what we're going to do? We're going to pump up a hydrogen. And I'm sorry I drew that weird, y'all, but y'all see what's happening, right? You see that we're building up hydrogens in this inner membrane space. Y'all see that? Even though I screwed up my drawing. Okay. Okay, now, hold that thought. So right now, what I want you to recognize is we used the energy of these electrons to create a situation where we pumped hydrogens into this inner membrane space. You see how many there are? Bunches of hydrogens. Incidentally, that makes this inner membrane space highly basic or acidic. Which one? Acidic, remember? Protons equal acid. Y'all see how that works? So this is an acidic environment now because we've got a whole bunch of protons, of hydrogen ions pumped in here. Y'all with me? Okay. The next thing I want you guys to notice is at this point, if I've drawn it correctly, and it looks like I kind of did, you see how I have this kind of up here going down, and then I had drawn this one a little bit lower? The reason I did that is I want to indicate to you that every time we move that electron around, it's losing a little bit of its energy, okay? By the time we've passed it 3 times, this poor guy is exhausted, but he still exists, and we have to put him somewhere. Where did I say we're going to put him? Oxygen, that's exactly right. So basically the way we do that is it's going to come off and it's going to dump onto oxygen, okay? So you have to have oxygen present in order to accept this electron, okay? Now, what happens then is you end up with actually I shouldn't put O2, you end up with 1 oxygen molecule that then you add 2 hydrogens that are in here, you add it to 2 of them, and you make metabolic water, okay? Does that make sense? We take this electron, we add it, I shouldn't put O2, we add it to the oxygen, that oxygen then joins some of the hydrogens here to make metabolic water. Yeah, so far? Not if you're with me, so I know. This is hard stuff. I promise you, you're like with me now, and you're gonna go home and you're gonna be like, what in the hell did she say? So make sure you're watching because I go over this in the video as well, okay? All right, so far we've used up our electrons' energy. We've made a gradient of protons. What are we gonna do now? So again, I drew this a little bit wrong. I should have drawn it here. So this guy's this little Christmas bulb. Let me see if I can actually erase. Let's erase this Christmas bulb and draw it right so y'all can see what's happening. All right. Sorry about that. Let me fix him. All right, the Christmas bulb should be here. Okay, now what's gonna happen is this Christmas bulb is this very cool enzyme called ATP synthase. Anytime you see a molecule, did I talk about this during chapter 8? Anytime you see a molecule that has ASE as the suffix, it is an enzyme, any and every time. If there's an enzyme called catalase, ATP synthase. We're going to learn about one called NADPH reductase. If you see ASE at the end of something, what is it, guys? Enzyme. Excellent. Okay, so we have all of these bad cats, all of these protons that have been built up. We now have a gradient, right? We have created a situation where we have a high concentration of protons here, and we've made a low concentration here because we keep pumping stuff out, right? So now we can take advantage of this electrochemical gradient. What's going to happen is these guys flow through this ATP synthase, and ATP synthase is the coolest little molecule. It's a tiny little molecular motor. What happens is it's almost like an electrical flow. As these positive charges flow, there's 2 subunits, one that's kind of square and one that's kind of rounded. It really does look like a molecular Christmas bulb. this part will rotate. So as these guys flow through, this thing spins, and that rotation, what that does is it takes ADP plus a phosphate group, so adenosine diphosphate plus a phosphate group, and guess what it makes, y'all? ATP, okay? Makes a lot of ATP. Are y'all with me so far? Now I've just got one more. If you understood all of this, you are cooking with gas. There's one more thing I just want to go back. Know how I had you guys skip this one right here? That's because this is where the FADH2 comes in. It works the exact same way. It just comes in a little farther down the chain, okay? So you end up getting a little bit less energy out of the electrons from FADH2 than you do from NADH. Okay? That's the only reason I wanted to bring that up. Do y'all have questions about this? In the video, I go through like an accounting of how you end up with a bunch of different ATPs. But what I want you guys to know is that if we go back here, you had made already 1, 2, 3, 4 ATPs. Oops. What you're going to make out of this is you're going to make 34 ATPs out of this process, plus the 4 that you had from previous processes. This process gives you 38 ATP per molecule of glucose. That's pretty awesome. Now, in the video, I tell you how this is a theoretical number. It turns out that we never actually are able to measure that, and the video explains why, but theoretically, you end up with 38 ATP. And you can only do that because you had a place to put your used up electrons, and that place was oxygen. I can tell you that one of the things we will ask you when you're seniors, if you are a senior in biology, we will ask you a bunch of different questions to see what you remember as a biology graduate from FMU. This question shows up all the time on our exit exam, which is, what is the purpose of oxygen? And it's a multiple choice question, so you don't have to remember it off the top of your head, but you should know that it acts as the final acceptor of electrons in the electron transport chain, okay? Yes, I'm right on time. Okay, now, this is cellular respiration. You guys watch fermentation. It's so much easier. So many fewer steps, okay? This is a lot more steps, but for a lot higher payoff. Now, let's contrast this with photosynthesis. So in this part of the chapters, we've used up chemical energy. Now let's see how we can make some chemical energy. So let's kind of switch our mindset. And I want to show you guys something. I'm going to write out a chemical equation for you guys. All right, so for this first chemical equation, I'm going to write C6H12O6 plus 6H2O. And that's going to give us 6O2 plus 6CO2. And then I'm just going to put plus ATP. Does anyone know what I just wrote a chemical equation for? Glucose, yeah. The whole thing is cellular respiration. Do y'all see that? This is actually the chemical formula for glucose. And sorry, I put water. I should have put oxygen. Y'all, I'm just not on my A game today. 6O2, and it gives you, yeah, there we go. Okay, so we're going to use glucose in cellular respiration. We're going to use oxygen in cellular respiration. Once you karate chop up your glucose, you end up with 6 waters, 6 CO2s, plus a crap ton of ATP, right? Now check this out. Y'all see where I'm going? What is this the chemical equation for? Photosynthesis. So can you see that photosynthesis kind of chemically is the reverse of cellular respiration and vice versa? You're getting a little bit different energy sources or energy outcomes. So here we have to have some light energy input. Here we're getting out some chemical energy, but they're basically the reverse of one another, aren't they? Spam caller. Go away, spam caller. Okay, another thing I want to point out to you guys, the waste product of cellular respiration is carbon dioxide. What's the waste product of photosynthesis? Oxygen. This is actually how we ended up with oxygen in our atmosphere. Okay? So the way we got oxygen in our atmosphere is just by this process happening over and over and over again, and it basically built up to where, and then that allowed for life to really proliferate, because you can see that once you have oxygen, you can do a lot of cellular respiration. You can do a lot of cellular respiration, you can make a lot of ATP, and you can be a lot of different kinds of critters and plants and things, okay? So let's think about photosynthesis. Let's see here. Let's think about photosynthesis in a broad conceptual sense, okay? You know how I said, okay, cellular respiration happens in 4 parts? Photosynthesis happens in 2 parts. So it really should be simpler. And I'm here to tell you that it's a little simpler. You're gonna say you are a lying liar who lies, Dr. Jones, but it really is. I think that the hurdle is that plants are so unfamiliar to us, okay? So let's think about this in a broad sense. First of all, you gotta start out with the daggum sun, right? Sunlight. So that's the sun, easy enough. The 2 parts that I want you to know are the parts of photosynthesis are what we refer to as the light capturing reactions. So I'm going to put light cap reactions. Is that okay? And then we're also going to have remember how we had the Krebs cycle or the citric acid cycle last time? We're going to have another biochemical cycle. This one is called the Calvin cycle. Okay, so I'm going to just draw something like this. Okay, this is called the Calvin cycle, named for Dr. Calvin, who discovered it. But it's also referred to as the light independent reactions. So you can immediately tell that like sunlight is necessary for this first half of the process, not necessary for the second half of the process. Now let's, just like we did last time, let's think about what the inputs and outputs are. Remember how I was like, oh, we put in glucose, we get 2 molecules of pyruvate, you get 2 ATP, blah, blah, blah. So let's think about what we're gonna put into this and what we're gonna get out of it as byproducts, okay? Into this, obviously we're going to put sunlight, right? So you're gonna put in light. Yeah. We're also gonna put in some water. You guys know plants need water. Evidently, I don't know that because I keep killing them. We're going to use up a little bit of, we're going to put in ADP and some inorganic phosphate, because we're going to be making a little bit of ATP in this first part, okay? And then we're going to use a molecule that's going to look really similar to you guys. Remember how we had NAD last time? And we turned it into NADH as our high energy electron carrier? We have a very similar molecule that gets used in plants. And it's going to be easy for you to remember. Here's why. This molecule is NADP. So there's a phosphate group added, but I always just remember there's a P in there, P for plants, P for photosynthesis, okay? So these are going to be our inputs into these light capturing reactions. What are we going to get out of these reactions? Well, the first thing you're going to get out of these reactions is some oxygen waste. And that is given off as gas, and we like that because we need that oxygen, okay? The next thing you're going to get out of this is, see how I have ADP plus PI? So we're going to turn that into ATP. The next thing we're going to do is we're going to take this NADP and we're going to reduce it to make NADPH. See how that hydrogen got stuck on there? That means that there's an electron on there. That means it's been reduced because reduction is gain, and it's got energy. All right, now what we're going to do, this is not a major pathway for making ATP. That is cellular respiration. In this process, we are making just enough energy to do the second half of the process. So this ATP and this NADPH exist to drive the chemical reactions of the Calvin cycle. What does the Calvin cycle do? The Calvin cycle uses this energy and it uses carbons that it gets from carbon dioxide in the atmosphere. So there's your carbon dioxide in the atmosphere. It's going to use these carbon dioxides from the atmosphere and this energy to generate sugar, okay? It's actually going to generate some little 3-carbon sugars. So I'm going to put 3C sugars. And how many sugars does glucose have? 6. So you actually, what this process does is you have to like run this twice. So I'll put 2X and that'll give you glucose. Are y'all with me on the big picture so far? So I've got about 15 minutes. I want to do one more thing with y'all before I turn you loose. So this isn't too bad, right? Now, I'm going to tell you right now, I don't think if I have time, I'll start on the Calvin cycle today. I do explain the Calvin cycle in the video. Some things that you'll need to watch out for, and I am going to post discussion questions. I know I haven't done that. I keep forgetting. I'm going to post those today. So you'll have discussion questions from our last lecture. That'll be in last week's folder. And you'll have discussion questions from this lecture in this week's folder. Again, if you were someone that was paying attention, those discussion questions were super helpful on your exam, right? They're almost like a study guide. They're not everything you need to know, but they help you to check your understanding along the way, okay? So one of the things I want to make sure that you guys get on your own, and I'll put in the study questions, make sure that you understand the Calvin cycle to the extent that I teach it. The book goes into a ton of detail. I'm not going to go into a ton of detail. But I do want you to understand there's one enzyme I want you to know in the Calvin cycle called rubisco. And that's an abbreviation because it has that ACEs at the end of every enzyme. Okay, we're not going to make you learn the whole name. Rubisco is an abbreviation. Okay. The other thing I want you to know is that the Calvin cycle has to turn multiple times to regenerate itself, and I want to make sure that you understand that. Okay, so those are things to look out for. But what I want us to focus in on for today, while we're together, is these light capturing reactions, and the reason is people get confused by them. But they're basically like the electron transport chain reactions of cellular respiration. You're using them for a different purpose, okay? So when you hear the electron transport chain, when you hear that term, people usually think cellular respiration, but there's an electron transport chain in photosynthesis as well. And if you understand one, then it's pretty easy to understand the other, okay? So let's look at it really quickly. First of all, let's get our geography squared away. Okay, so anyone know where photosynthesis occurs in plant cells? Like what part of the plant cell it occurs? Chloroplast, yeah, excellent. Several of y'all know. So just like I drew you a little mitochondria, let me draw you a little chloroplast so y'all know where we are, okay? So here is a little chloroplast. I'm just gonna put it up here so I have room elsewhere. Just like our mitochondria, it's going to be double membraned, okay? So there's an outer membrane here and an inner membrane here. Incidentally, this is another organelle. It's part of that endosymbiotic theory. Make sure that you understand what that is, okay? If you were to look at one of these under the microscope, you would see that the interior of these guys is organized with these little stacks of discs, okay? I drew this. They're about the same size. They don't look like snowmen. They're about the same size, okay? Each stack is called a granum. That's singular. If you had a whole bunch of stacks together, the plural would be grana, okay? So if you see grana versus granum, don't get confused. We're just talking about the stacks, okay? If you look at one of these little discs that makes up these stacks, one little disc, just one of them, is called the thylakoid, and this is where the action is going to occur. So the thylakoid has a membrane that I've drawn here, and it has an open up inside part. The open inside part of the thylakoid is called the thylakoid, and I'm going to abbreviate that THY, lumen. Okay, good so far, yeah? Then you have the stuff out here. This is called the stroma. So now you guys kind of know where you are, right? So let's draw what the light reactions of photosynthesis look like, okay? So we are going to draw for ourselves. I'm going to draw it correctly this time. Haha. Okay, so out here is the cytosol of the plant cell. This is the outer membrane of the chloroplast. This is the inner membrane of the this is the outer membrane of the thylakoid. I lied to you guys. This is the stroma. Here's where we are, guys. We're embedded in here. Sorry. Y'all with me? Sorry, I hate it when I say something wrong and then I have to correct myself, but you're good? Kelsey, you good? Okay. So there's the stroma. This is the outer membrane of the thylakoid. This is the inner membrane of the thylakoid. That makes this the lumen of the thylakoid. All right, let's do what we did last time. Let's draw ourselves some little protein complexes, but I'm going to draw them correctly this time. So I'm going to draw one kind of here, and I'm going to draw one here, and I'm going to draw one here, and I'm going to draw our Christmas bulb, but I'm going to invert it, y'all. Here's our Christmas bulb. See how I turned it upside down from where it was last time? Okay. This is the same enzyme. This is still ATP synthase. But it's upside down, and you're going to see why in just a second, okay? So what happens in photosynthesis? We need a source of electrons. And what we're going to use as our source of electrons initially is going to come from our pigments. I go into a whole thing about pigments. Chlorophyll is a pigment. So our electron source is our pigments. And what happens is light comes in, and light energy comes in the form of something called photons. Have you guys heard of photons before? Yeah? So we're going to actually pretend that 4 photons of energy, 4 little particles of light have hit our pigment. And what that does is it's going to excite, it's going to strip off 4 electrons. Are y'all with me so far? We're going to get 4 electrons off of this pigment center here. We're going to use these 4 electrons. And remember, electrons equal energy, right? Okay. So we have these 4 electrons. These 4 electrons are going to hitch a ride on a shuttle bus. Does this sound familiar to you? It should. Okay. So first off, this shuttle bus has a different name. It's called PQ for plastoquinone. Again, I'm not interested in names as much as I'm interested in concepts. So these 4 electrons, they're going to hitch a ride onto this shuttle bus. Shuttle bus drops them off. Here's the shuttle bus. Here's our 4 electrons. We're going to use that movement of the electrons to, guess what? Pump hydrogens, but this time we're pumping them from here to here. Do y'all see that we're doing this a little upside down? But otherwise, do you see how this is the same? We're just using light to generate our electrons because we're using light to strip them off of the pigment molecules, and we're just passing them over, okay? All right, now we're going to, just like last time, these guys are going to catch a second bus. They're going to catch a second shuttle bus. This second shuttle bus is called plastocyanin. Don't worry about it, but I will label it for you guys, PC. And we're going to drop these guys off here. Bada boom. Let's see here. So here's our PC. And here's our 4 electrons. Okay. Once again, guess what? We're going to use that to pump hydrogens. Bada bing, bada boom. Y'all with me? Okay. Now, at this point, what we've done is we've pumped a bunch of hydrogens in here. And notice how I took our electrons. They had been what we call photoexcited. They had been given energy by the sun, and they had a lot of energy, and we passed them, and then they had some energy, and we passed them, and see how I made that lower each time? Now they're exhausted. But we want to use them one more time, okay? So what happens is more light comes in. It's going to give these guys some rejuvenated energy. That energy is going to hitch it to a 3rd shuttle bus. This shuttle bus is called ferredoxin, and I'm just going to abbreviate it FR. That shuttle bus is going to pass it to one of the only enzymes I'm going to expect you guys to know, okay? That shuttle bus passes this to an enzyme called NADPH reductase. So let me write that down. NR, I'll put it up here. NR equals NADP reductase. So guess what NADP reductase does? it reduces NADP. So you get a molecule of NADP that's hanging around in here in this region, and you add a hydrogen to it. And bear in mind that I've kind of got it out here, but this is all happening in this inner membrane space, okay? Now what you've done, you've generated an electron carrier, you generated an energy carrier, okay? So this guy is going to, let me use this for just a second. This guy is going to get used later in the Calvin cycle as an energy source. We good so far? Now, the next cool thing, we've also built up a gradient here. If you do this over and over again, you're going to end up with a whole bunch of hydrogen ions, right? These hydrogen ions are going to have a high concentration here. They can flow through our ATP synthase. When they do, it causes this thing to turn and twirl, and it causes us to take ADP plus a phosphate and turn it into ATP. This is the second thing that we can then use to drive the Calvin cycle. Do y'all see how that's related? Do you see how similar that is? That's why I teach these two things together, okay? So one more thing that we need to do to complete this, to complete these reactions. And that thing is, we lost 4 electrons. If y'all go back to the very beginning, I told you that light came in here and it caused 4 electrons to come off this guy, didn't it? That means there's 4 electrons missing, right? So let's just take some water. Because remember how I told you all I needed water for this? This is why plants need water. We're going to take 2 molecules of water, and we're going to split that. And that's going to give us 4 hydrogens plus 2 oxygens. Do y'all see that? If I split that water, what happens? So we can then take these 4 hydrogens, take the electrons from these 4 hydrogens, add them back here. Do y'all see how I did that? We took the electrons from these hydrogens, added them here. That gives us 4 more H pluses to add to this gradient, right? And you end up with this extra oxygen that the plant doesn't need. So there's your oxygen waste that goes bye-bye. So if we go back here, I told you needed water, adenosine diphosphate, NADP. Let's go here. Here's where your water is. Here's where your adenosine diphosphate is. And here's where your NADP is, right? I told you what you were going to get out was oxygen, ATP, and NADPH. Bada boom, we got oxygen here. We got NADPH, and we got ATP. I don't lie. Told you the truth. Okay, now these guys will go into the Calvin cycle, which I actually, is my clock, is it 11:02? Are we good on time? We did it 11:10, right? I'm gonna real quick, just give you a quick preview of the Calvin cycle. I know you guys are tired, but I only get you once a week. So suck it up. All right, one last thing, and then I'll leave you guys alone because we do have an exam coming up. All right, let's talk Calvin cycle for a second. So if we go to our roadmap of photosynthesis, okay? Here's our roadmap, and I already taught you guys this roadmap part here, right? Now let's look at this part here. All right, how's that gonna work? I'm just gonna give you the broad strokes of that, okay? So let's draw ourselves a little cycle here, okay? And I'm gonna leave some spaces. And okay, so this whole process of the Calvin cycle starts out with a 5-carbon sugar. So I'm gonna draw 1, 2, 3, 4, 5. Do y'all remember in the Krebs cycle or the citric acid cycle that I just showed you? We started out with 4 carbons that just kept getting used over and over again. They kept getting recycled. This is the part that keeps getting recycled, okay? Y'all with me so far? This sugar is called RUB-P, which stands for ribulose bisphosphate. Just know RUB-P, that's easier, okay? And the reason it's called ribulose bisphosphate is there's a phosphate group on either end, but the circles are the carbons. There's 1, 2, 3, 4, 5 carbons. Now, what's gonna happen is the plants have this awesome ability to take CO2 from the atmosphere. How many carbons are in 1 molecule of CO2, guys? 1, do you see that? There's just the 1 carbon. So we're gonna tack that carbon onto here. So it'll be 1, 2, 3, 4, 5, 6. Okay, and there's still a phosphate here and a phosphate here. But do y'all see how I just added that carbon here? The enzyme that does this is a really important enzyme. It is called rubisco. Again, this is an abbreviation. I give you more information about it in the video. It's an important enzyme. It's the most abundant enzyme on the planet, the most abundant protein on the planet, because you can imagine we need lots of photosynthesis, right, to support life. So it's a big deal kind of enzyme. But what rubisco does is we say it fixes carbon. In other words, it adds carbon to these molecules. So it's basically what is creating sugars. Then what's going to happen is kind of like glycolysis, we're going to end up with this thing getting broken in half. So you'll end up with something that looks like this. 1, 2, 3. And then there'll be a 1, 2, 3. And then you're going to go through a series of manipulations, just like I said you did in the other citric acid cycle. These manipulations are going to require some energy. So basically the ATP that we generated in our light capturing reactions, we're going to use that up, and that'll regenerate our ADP and our phosphate. The NADPH that we made in our earlier reactions, we're going to strip off that electron, we're going to use it up, and we're going to use that energy to drive these reactions. At the end of all of this, what we've done, it's doing some manipulation, some chemical manipulations, and you end up with something that looks like this. It's a 3-carbon sugar, boop, 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 and there's a hydrogen on this one, and there's a 3-carbon sugar, boop, 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 and a hydrogen on this one. This is called glyceraldehyde 3-phosphate, or G3P. This is that 3-carbon sugar that you get, okay? That 3-carbon sugar, and remember how I said you get a 3-carbon sugar and you need 2 of them to make a glucose? This is that one part of that glucose, okay? The other one stays in this cycle. But here's the problem. And the problem is this. How many are left? How many carbons are left? 1, 2, 3. Y'all see that? We took these 3 out of the cycle. We have 3 left, right? But we need to regenerate our starting product that had 5. How the plants do this is they're running a whole bunch of these reactions at once. So let's look at this. I'm going to say 1, 2, 3. Okay, each one of these represents a turn of the Calvin cycle. The first time you do the Calvin cycle, you get 2 G3P molecules, right? So you get a 3-carbon sugar and a 3-carbon sugar. Y'all with me so far? One of these is going to be stripped off to make glucose. Do y'all believe me so far? All right, second turn, we're going to get a 3-carbon sugar and a 3-carbon sugar. And then third turn, yeah, right? If we add all these carbons up, what is 5 times 3? Right? So that's 3, 6, 9, 12, 15. Yeah? Okay, well, again, that means that if we divide this by 3, we get a 5. Let's go back here. How many carbons were there here? 1, 2, 3, 4, 5. If we run this thing 3 times, we can regenerate 3 molecules of a 5-carbon sugar. Yes, ma'am?
So each one of the spins does not go towards the glucose?
Not each one goes toward the glucose, 1 and 3.
Okay.
Actually 1 and 6, but 1/2 of the, yes, exactly.
Okay.
Do y'all see how that works? And obviously there's some more chemical manipulation that has to happen to get you back to that 5-carbon sugar, but that's kind of how you end up with that. Yes, ma'am. So you end up with 3 molecules of a 5-carbon sugar, because if we go back here, bada bing, remember that this was 5 carbons, 1, 2, 3, 4, 5, and then we turned this thing and we have a 3-carbon molecule. We got to get back to 5. Okay, so you have to run it 3 times in order to be able to make the math work out, so that you can bring it back to the product. Okay, so that once you do that, you get it with 3 of these guys, and you can run the cycle again. Okay, guys, that is a super quick and dirty, but the thing is, I'm hoping that if you guys see how those are related, now go back, watch those lecture videos, and you'll be like, oh, now this makes sense. But remember, I'm building the Christmas tree. It's your job to decorate it by watching my longer lectures. And don't forget that you have a homework due tomorrow by 5 p.m. Also, don't forget that you have your discussion thread that you can get extra credit. A lot of people did really well in the exam because they got themselves sweet, sweet points. All right? Okay, go away. Talk to you later.